The third big RNA processing event that happens in our cells is RNA splicing. In RNA splicing, specific parts of the pre-mRNA called introns are recognized and removed by a protein and RNA complex called the spliceosome. Introns can be viewed as junk sequences that must be cut out so the good parts version of the RNA molecule can be assembled. Don't forget the term spliceosome. We'll be learning about it later. So, this piece of the RNA that are not chopped out are called exons. The exons are pasted together by the spliceosome to make the final mature mRNA that is shipped out of the nucleus. So, it will be, moving out from the nucleus. Later, some regions, this is a portion of DNA, 5' to 3' orientation is shown. Exon portions are there. Exons in the sense coding sequences. Then intron portions in the middle of two exons, two intron portions are shown. So these intron portions are non-coding sequences often referred to as junk DNA portions. So these portions should be removed in case of transcription. So just remove these portions with the help of a process called splicing to produce a mature mRNA. Diagram of pre-mRNA showing exons and introns. Along the length of the mRNA, there is an alternative pattern of exons and introns. Exon 1, intron 1, exon 2, intron 2, exon 3. Each consists of a stretch of RNA nucleotide. During splicing, the introns are removed from the pre-mRNA and the exons are stuck together to form a mature mRNA that does not contain the intron sequence. A key point here is that it is only the exons of a gene that encode a protein. Not only do the introns not carry information to build a protein, they actually have to be removed in order for the mRNA to encode a protein with the right sequence. If the spliceosomes fail to remove an intron, an mRNA with an extra junk in it will be made and a wrong protein will get produced during translation. So their removal is very essential. That is very important case of alternative splicing. So alternative splice, why splice? We don't know for sure why splicing exists, and in some ways it seems like a wasteful system. However, splicing does allow for a process called alternative splicing in which more than one mRNA can be made from the same gene. Through alternative splicing, we and other eukaryotes can sneakily encode more different proteins than we have genes in our DNA. In alternative splicing, one pre-mRNA may be spliced in either of two or sometimes many more than two different ways. For example, in the diagram below, the same pre-mRNA can be spliced in three different ways depending on which exons are kept. This results in three different mature mRNAs, each of which translates into a protein with a different structure. So exon 1, exon 2, exon 3, exon 4, exon 5 regions are there. And RNA, exon 1, exon 2, exon 3, exon 4, and exon 5 regions are there. This will be undergoing alternative splicing to create mRNA translation. Their arrangement is different. Look how their arrangement is way more different. Their position is different, like that. Just arrangement is different. So alternative splicing will be taking place. So mRNA is there. Translation is happening in this mRNA portion. So later, a different protein is formed in each case, but their sequences are to a great extent similar. Their DNAs are translated into a protein structure. Okay, DNA is similar, but their arrangement after alternative splicing is different. Their non-coding portions are, removed from this, and their translation produce proteins with a different arrangement: protein A, protein B, and protein C. Diagram of alternative splicing: a sequence of DNA encodes a pre-mRNA transcript that contains 5 regions that may potentially be used as exons. A sequence of DNA encodes a pre-mRNA transcript that contains 5 regions that may potentially be used as exon 1, exon 2, exon 3, exon 4, and exon 5. These exons are arranged in linear order along the pre-mRNA and have introns in between them. In splicing event 1, all 5 exons are retained in the mature mRNA. It consists of exon 1, exon 2, exon 3, exon 4, and exon 5. When it is translated, it specifies protein A, a protein with 5 domains. Coil 1 specified by exon 1, coil 2 specified by exon 2, just look at this figure. Loop 3 specified by exon 3, loop 4 specified by exon 4, and coil 5 specified by exon 5, like that. In splicing event 2, exon 3 is not included in the mature mRNA. That is the difference. It consists of exon 1, exon 2, exon 4. 3 is absent there. Exon 5. When it is translated, it specifies protein B, a protein with 4 domains. Coil 1 specified by exon 1, coil 2 specified by exon 2, loop 4 specified by exon 4, and coil 5 specified by exon 5. It does not contain loop 3 because exon 3 is not present in the mRNA. In splicing event 3, exon 4 is not included in the mature mRNA. It consists of exon 1, exon 2, exon 3, and exon 5. When it is translated, it specifies protein C, a protein with 4 domains. Coil 1 specified by exon 1, coil 2 specified by exon 2, coil loop 3 specified by exon 3, and coil 5 specified by exon 5. It does not contain loop 4 because exon 4 is not present in the mRNA. Then, genetic information transcribed from DNA molecules contains both introns and exons. But for mRNA to be translation ready, it must only have exons. So, we have to splice these introns out and keep only exons. This process of removing introns and keeping only exons in the mRNA sequence is called mRNA splicing. The process of splicing occurs by recognizing a specific sequence that initiates the cleaving out of the introns and rejoining of exons with utmost precision and accuracy. The splicing process is made possible by complexes of proteins and RNA molecules called spliceosomes. Each of the spliceosomes consists of 5 subunits called snRNPs for small nuclear ribonucleoproteins particles or riboparticles. Small nuclear ribonucleoparticles. These snRNPs are only found in the nucleus. These spliceosomes recognize sequences at the 5' end and 3' end of the intron. The specific sequence, they recognize the GU nucleotide at the start of 5' end and AG nucleotide at the end of 3' end. Now that these spliceosomes recognize the introns, they cleave them out by breaking the sugar phosphate of pre-mRNA at the 5' end of introns and covalently attaching it to a nucleotide internally within the intron. This results in cleaving out of introns. Spliceosomes now connect the free 3' end of one exon to the adjoining exon on a free 5' end, thus making the joining possible. Now the mRNA that we have in a mature mRNA with the 5' cap, 3' poly A tail and only exons coding sequences. Okay, here we could see a picture which depicts all this information we just shared with you. I will be showing this picture in large type. So look at this portion. Splicing is taking place. Exon 1, 5' splice site, intron, branch point, polypyrimidine tract, 3' splice site, UT and UTAF, transesterification, then lariat intermediate, exon 2, transesterification to U2, U5 and U6, lariat intron, like that. Transesterification is also taking place, just remember that. So this is about splicing, how exon 1 is there, how 5' splice site is created and 3' splice site is created, and there is a structure called spliceosome, etc. are very important, and how their arrangement differs when one single DNA sequence is there that contain many exons and introns. But after this alternative splicing, various kinds of proteins are produced with the help of that alternative splicing. Thank you for listening.